Welcome back to the deep dive. Today, we're plunging into a topic that might feel a bit daunting, but honestly, it's essential. The world of emerging infectious diseases.
Yeah, absolutely.
We're not just looking at the pathogens, the bugs themselves, but how these huge global forces, like climate change especially, are really shaking up the risk landscape.
Right.
And then we'll look at the other side, the science, the counteroffensive, some surprising ways researchers are fighting back, looking at, well, everything from foundational work like gene therapy ideas to the really cutting-edge surveillance tech.
Mm-hmm.
We've pulled together quite a stack of sources for this one, research papers, big reports from places like PubMed, NIH, the WHO, plus insights from researchers at places like Harvard.
It's a lot of material.
It is. And our goal here is really to cut through that complexity, pull out the key takeaways for you.
Yeah, our mission here is pretty straightforward.
Hmm.
We want to help you get informed on something that, let's face it, touches every single person's life. We'll unpack the interconnected factors driving the risks and also show you the many different efforts happening to, predict, prevent, and treat outbreaks. Think of this as your shortcut to getting a handle on a really critical global challenge.
Okay, let's dive in then. When we think about infectious diseases, I think often the focus jumps straight to the big pandemics, the ones that grab headlines.
Right, the headline stuff.
But the sources really emphasize this constant background noise, this drumbeat of infectious diseases that goes way beyond those major events. Is that fair?
That's absolutely right. And we use this term, emerging infectious diseases, EIDs, basically for pathogens that are either totally new to humans or maybe ones we knew about that are suddenly spreading more easily or, popping up in new places.
And history shows these outbreaks are just, well, incredibly frequent.
Oh, completely. And the scale can be just staggering. I mean, look at the 1918 influenza. It infected maybe 500 million people across the globe.
500 million.
Yeah, causing maybe 50 million deaths. It's still the biggest EID event we've seen in the modern medical era. And more recently, you've got SARS, MERS, swine flu, Ebola, chikungunya, Zika, obviously SARS-CoV-2.
Right.
And don't forget diseases like HIV, which, emerged decades ago from a spillover. It's infected over 85 million people worldwide.
Wow. And even with all the amazing progress in vaccines and treatments, the NIH data we looked at said something like 23 million Americans still need treatment for infections every single year.
Yeah, that number really drives home that constant drumbeat idea. It's not just about the pandemics that make the news.
Okay.
You've got ongoing killers like tuberculosis, malaria, diarrheal diseases, pneumonia. These cause huge numbers of deaths globally, year in, year out, especially hitting vulnerable populations hard.
So what's turning up the volume on this drumbeat? What are the big forces driving this increasing risk for EIs?
Well, the sources point to a few key interconnected factors. First, just sheer population growth, coupled with way more human mobility.
More people moving around more.
Exactly. Simple as that, really. Helps pathogens spread further, faster. Then there's the impact of pollution, environmental degradation that can weaken ecosystems, sure, but also human health, potentially making us more susceptible.
Makes sense.
And the global population is aging. That's another factor, because older individuals often have weaker immune systems, making them more vulnerable.
And layered on top of all that is something the sources highlight as a major, really urgent concern: antimicrobial resistance, AMR.
Yes, AMR it's a massive global threat. It's essentially where the microbes, bacteria, viruses, fungi, parasites, they evolve defenses against the drugs we use to kill them, like antibiotics becoming useless.
So common infections become untreatable again.
Potentially, yes.
Yeah.
Or much, much harder to treat. It's basically an evolutionary arms race we're in.
Okay, that gives us a clear picture of the sort of baseline risk. Now let's get into where it gets, I think, really interesting and maybe quite concerning, this deep link between climate change and zoonotic spillover.
Right, zoonotic spillover. This is absolutely central. It's when a pathogen makes the leap, crosses the species barrier from a non-human animal into humans.
And the numbers are striking, aren't they? Like most human diseases started this way.
That's what the estimates suggest, yeah. Somewhere between 60, maybe even 75% of all human infectious diseases originally came from animals.
So the vast majority of things that make us sick were circulating in wildlife or maybe livestock first.
That's the idea. And climate change isn't just sitting in the background here. It's actively, pouring fuel on the fire, accelerating this spillover risk.
How does climate change specifically drive this?
It works in a few ways. Changing temperatures, different rainfall patterns, more extreme weather. These things directly mess with animal behavior and where they live.
Okay.
For instance, if warmer weather or changes in plants cause a sudden boom in the food for, say, deer mice in the American Southwest,
Hmm.
well, their populations can explode.
Right.
And that means more potential contact with humans. That exact dynamic was linked to a hantavirus outbreak back in the 90s. Climate shifts can also force animals to move, maybe search for water or food in new areas they wouldn't normally go to.
So climate change is kind of pushing wildlife and humans closer together. Is that it?
In many cases, yes, that's a big part of it. When ecosystems get stressed, maybe from heat, drought, deforestation, animals might be forced to look for water or food near our towns, near our farms.
Increasing the chances for contact.
Dramatically increases the opportunities for interaction and, yeah, for spillover. The sources mention the Australian black flying fox. It carries Hendra virus. Its migration patterns have shifted, probably because climate change affected its food sources, bringing it into contact with horses and humans further south than before.
Wow.
And climate change is also linked to these big atmospheric patterns like El Nino, which can then influence how diseases like cholera spread geographically.
And our own activities, human activities, often tangled up with these environmental changes, they also play a huge part in spillover, right?
Oh, absolutely. Direct contact with wildlife is a big one. Things like hunting, trading, eating wild animal meat, sometimes called bushmeat, puts people right up close with animal blood, tissues. It's a clear path for pathogens.
Like HIV, didn't that start that way?
The evidence strongly suggests HIV's emergence is linked to handling primate meat, yes.
And wet markets. We heard so much about those with COVID-19.
Yeah.
How do they fit?
Right, wet markets. They can be major amplifiers. You often have many different species crammed together in cages, stressed, waiting to be sold.
Creates a perfect storm.
Scientists call them mixing vessels. It's like the perfect environment for viruses to jump between species, maybe even recombine into new, dangerous forms, and then potentially infect the humans working there or shopping there. We've seen it happen with flu viruses in pigs in similar settings, and yeah, SARS-CoV-2 is widely suspected to have emerged from that kind of environment.
And just changing the land itself. Building roads, cutting down forests.
Exactly. Land use change is critical. Deforestation, expanding farms, building settlements deeper into natural areas. It destroys habitats, yes, but it also directly forces more contact between us, our livestock, and wildlife that might be carrying pathogens.
And there are specific examples.
Yeah. Sources point to research linking, for instance, forest fragmentation in parts of Africa with a higher risk of Ebola outbreaks.
Huh.
And this loss of habitat connects to biodiversity loss in another interesting way, something called the dilution effect.
Dilution effect. What's that?
The idea, based on research, is that in really diverse ecosystems with lots of different animal species, a pathogen might infect many different types of animals, but maybe not become super common in any one of them.
Yeah.
Especially not the ones most likely to transmit it to humans.
Okay.
So high biodiversity can kind of dilute the pathogen's prevalence in the key reservoir hosts, lowering the overall risk of spillover to us. But when we change the land, reduce biodiversity, we sometimes end up favoring species like certain types of rodents, for example, that happen to be really good hosts for particular pathogens.
So paradoxically, simplifying the ecosystem can concentrate the danger. Protecting nature actually helps protect us.
In this context, yes, preserving biodiversity seems to act as a kind of buffer. And, our own animals, our livestock and pets, they're part of this web too.
How so?
Well, they can be direct sources of pathogens that infect us, or they can act as sort of bridge hosts. They pick up a pathogen from wildlife, maybe it adapts a bit within them, and then they pass it on to humans. Or they act as mixing vessels themselves, like pigs with influenza. Even dogs, apparently, can sometimes act as bridges between wildlife and people where cities meet forests.
This whole picture just screams interconnectedness, human health, animal health, the environment.
Which brings us right to the One Health approach.
Right.
That perspective is totally foundational now in tackling these issues. One Health says, look, you can't separate human health from animal health or the health of the environment we all share. They're fundamentally linked.
You have to address all three together.
Exactly. You can't effectively handle future infectious disease threats otherwise.
Okay, so given all these risk factors, this complex web, the consensus from the sources we read seems pretty blunt. Another pandemic isn't just possible, it's basically inevitable.
Yeah.
It's not if, but when.
Unfortunately, based on the trends and the factors we've just discussed, that does seem to be the sober assessment.
Huh.
But look, the good news is we're not, starting from zero. We have learned some hard lessons, especially from COVID-19.
Yeah, we learned lessons.
And we do have new tools, new approaches.
What were some of the key lessons that stood out from the COVID experience?
Well, one big one was just how reactive we tend to be. Governments, people.
Hmm.
There's a real reluctance to invest seriously in pandemic preparedness before the crisis hits.
Yeah, wait till the house is on fire.
Kind of.
Oh.
Another lesson was just the sheer endurance needed. Responding to a major outbreak is a marathon, not a sprint. It takes sustained effort over a long time, and vaccination, once you have it, is just absolutely critical for control.
But the science moved incredibly fast, didn't it? That was maybe a more positive lesson.
Oh, remarkably fast. The speed of sequencing the virus, developing effective vaccines, it was genuinely unprecedented. Real hope there.
Yeah.
But the sources are quick to point out that capability doesn't just appear out of thin air. It depends entirely on long-term, sustained investment in basic science and the translational research that turns discoveries into actual tools.
You need both, and we saw new technologies really come to the fore during the response,
It
too.
did. Things like wastewater surveillance testing sewage to track virus levels in communities,
Hmm.
that became huge.
Right.
Advanced computer modeling to try and predict spread, using new kinds of data streams, mobile phone data, genomic data. These tools are much more developed and appreciated now. The CDC even set up a dedicated center for forecasting and outbreak analytics,
Integrating all that data.
trying to, yes. But interestingly, there were nuances even there. Some sources noted that these really complex big data models weren't always the most helpful thing for guiding responses in specific local situations.
Why not?
Because public health is fundamentally local.
Right.
You need data that's relevant and makes sense for that specific community, that context. Sometimes the big models were too abstract.
And the social side of things, that seemed like a huge challenge.
Yeah, the sources suggest that was poorly handled in many places. You saw stark inequalities globally in who got vaccines.
Definitely.
And the pandemic just amplified existing social and economic divides. Vulnerable populations were hit disproportionately hard, not just by the virus itself, but by the economic fallout, too.
And even the basics like masks, testing, ventilation. There seemed to be a lot of initial confusion or delay in getting those widely adopted.
There was. But the understanding of how important those basic non-pharmaceutical tools are grew a lot.
Mm.
Experts now think that going forward, things like masks, rapid tests, better building ventilation, they can and should be rolled out much faster and more decisively in that critical early phase before you have vaccines or treatments ready.
So we have these lessons. What are the ongoing hurdles to actually putting them into practice effectively next time?
Well, a big one is fragmentation. In some countries, there still isn't a single coherent national response plan. Responsibility is split across lots of different agencies, different levels of government,
Bureaucracy gets in the way.
and infectious diseases move way faster than bureaucratic timetables, unfortunately. Another huge hurdle is politics. Public health responses can become intensely politicized.
And we certainly saw that.
Yeah, and that makes collaboration incredibly difficult, erodes public trust. Building that trust back requires really clear communication, being honest about what we know, what we don't know, acknowledging the uncertainty.
So beyond just reacting better when something hits, what about getting ahead of it? Proactive prevention.
That's absolutely crucial. We need much better surveillance, robust systems for testing and tracking pathogens we already know about, but critically, also systems designed to spot new threats emerging early.
Early warning systems.
It's exactly better planning for how to respond quickly and flexibly, and fundamentally, investing in the basic public health infrastructure, the labs, the trained workforce that's essential but often gets neglected between crises. And looping back to that One Health idea, prevention isn't just about pills and shots. The sources emphasize that things like good governance, tackling social inequality, even ensuring basics like food security, these can be incredibly powerful tools against infectious disease.
How so? Like the trial in India.
Exactly. They mentioned the rations trial. It showed that giving nutritional support to households with TB patients actually had a protective effect against the disease spreading, an effect comparable to some vaccine candidates.
Wow. So just ensuring people have enough good food acts like a form of prevention. That's fascinating.
It really is. It shows how these broader social and environmental factors are genuinely part of the preparedness toolkit.
Okay, let's shift focus a bit now to the scientific arsenal itself. What are some of the novel technologies and therapeutic ideas the sources talk about for actually fighting EIDs?
Right. The science and tech front is moving incredibly fast. Lots of new ways to monitor, screen for, and potentially treat these diseases. One really critical area is genomic surveillance. This is basically reading the genetic code of pathogens as they spread.
Like tracking mutations.
Exactly. It lets us track how viruses like SARS-CoV-2 evolve, spot concerning variants, like B41.1.7, P.1, understand what mutations make them more transmissible or severe. It also helps spot drug resistance, say in bacteria like Salmonella. And all that genomic data directly feeds into designing better vaccines and drugs.
But it needs resources.
Big time. And crucially, it needs effective global data sharing to work properly.
So it's like real-time evolutionary tracking to predict the pathogen's next move.
That's a good way to put it. Then you have epidemiologic analysis and modeling. This uses data on cases, maybe contact tracing information, though, there are always privacy concerns there.
Sure.
And combines it with all sorts of other data, genomic, demographic, even climate data. Feeds it all into predictive models, often using AI and machine learning now.
What are these models used for?
During COVID, they helped inform vaccine rollout strategies, like deciding who to prioritize in Hong Kong. They also help guide where to put resources for diseases like dengue, Zika, West Nile. And networks like GeoSentinel, which tracks diseases in international travelers, provide vital early warning data too. Mobile applications are another fast-moving area. Apps for surveillance, letting people report symptoms, maybe track exposures. Some are even using AI for assisted diagnosis, like analyzing phone pictures to identify things like monkeypox lesions.
Like the health code system in China.
Yeah, that was an example, integrating personal health data for risk assessment. It showed the potential, but also flagged those significant privacy issues.
But yeah, the potential for using mobile tech for rapid surveillance, maybe early diagnosis, it's huge. We just need to figure out the data integration and privacy aspects carefully.
So our phones could become like sophisticated disease detectors. Potentially, yeah, if done right and responsibly.
Yeah.
Then there are biosensors. These are devices designed for rapid, cheap, easy-to-use testing. They basically combine something biological, like an antibody that grabs onto a virus particle, with a sensor that detects that interaction.
What do these look like? Wearables.
They can take many forms. People are working on wearable sensors, maybe in a patch or watch, that monitor things in your sweat or the fluid just under your skin.
Wow.
Or even ingestible sensors, tiny pills you swallow that could analyze things in your gut. Implantable sensors, too, for long-term monitoring inside the body. The promise is huge for continuous monitoring, catching infections really early, especially if you combine them with AI to analyze the data and wireless tech to send alerts. And finally, getting tests to where they're needed most. Point of care or POC tests.
Like rapid tests.
Exactly. Tests that give you results quickly, right there in the clinic, or maybe even at home. They allow for immediate diagnosis, faster treatment decisions, quicker isolation to stop spread. You've got protein tests, antibody tests, molecular tests like PCR, but faster. And now we're even seeing smart POC devices that can automatically transmit the results wirelessly, helping manage individual patients better and feeding into the bigger surveillance picture. One source mentioned a 5G-enabled sensor for SARS-CoV-2, for instance.
Okay, so that's the cutting edge of monitoring and diagnosis. But the sources also took a look back, didn't they? At more foundational therapeutic research, specifically some early work on gene therapy for infectious diseases. Seems like a bit of a jump, but I guess it shows the history here.
It does, and it's really valuable to see that history, that scientific lineage. The sources included discussions of research from, like, the late 1990s into gene therapy for infectious diseases.
Really? Gene therapy for infections back then?
Yeah. The specific technologies have obviously evolved massively since then, but the core ideas, the targets, they were already being explored, laying some groundwork. And HIV was really the main focus, the kind of paradigm disease for trying out these early gene therapy ideas.
What kinds of strategies were they trying back then for something like HIV using gene therapy?
Well, they were exploring several different angles. One was using things called antisense nucleic acids and ribozymes.
Okay, what are those designed to do?
So antisense molecules are basically small bits of genetic material designed to stick to specific sequences on the virus's own RNA or DNA. The idea is to physically block the virus from using its genes to make necessary proteins.
Gum up the works.
Pretty much. And ribozymes are different. They're special RNA molecules that act like tiny molecular scissors. You can design them to specifically find and cut the virus's RNA at critical points.
So disabling the virus's instructions?
Exactly. With both antisense and ribozymes, the goal was to stop the virus from replicating inside the cell, maybe even make the cell permanently resistant. But even back then, they knew there were challenges, like needing to get lots of these molecules into the cells and the virus mutating to escape the target. Researchers were designing ribozymes to hit key viral parts, trying to be strategic. Another approach was using something called transdominant negative proteins, or TNPs.
Yeah.
These are basically intentionally broken versions of essential viral proteins.
So, like putting a faulty part into the virus's assembly line.
That's a great analogy, yeah.
Yeah.
The idea is you make the cell produce these faulty viral proteins. They then compete with the virus's normal functional proteins and interfere with replication. For HIV, they looked at making faulty versions of key regulatory proteins called Rev and TAT.
How would that work?
The hope was these TMPs would maybe bind up cellular machinery the virus needs, or just generally disrupt the essential viral processes. They even explored combining different TNPs, thinking that might be more effective. One example mentioned was RevM10, a mutant Rev protein designed to mess with its cellular partner. TAT TNPs were designed to grab onto cellular transcription factors. There were even early clinical trials exploring these concepts. Then there were intracellular antibodies, or intrabodies.
Antibodies inside the cell. I thought they worked outside.
Normally they do. But these were antibody fragments engineered specifically to be produced and function inside cells. The idea was to trap or neutralize viral proteins right after they're made, before they can do their job or assemble into new virus particles. For HIV, they look at intrabodies targeting the ENV protein on the virus surface, or the Rev protein, trying to trap them in certain parts of the cell. Even intrabodies against the reverse transcriptase enzyme were studied. And finally, they were looking at gene-based immunotherapy.
Using genes to boost the immune system?
Kind of. One idea was to put genes into cells that would stimulate a stronger immune response against cells already infected by the virus, particularly getting cytotoxic T lymphocytes, or CTLs, involved. Those are the immune system's killer cells. Another angle was modifying a patient's own immune cells directly, what we now call adoptive cell therapy, engineering them with genes to make them better at finding and killing the virus. Again, early clinical trials explored these ideas for HIV and also for other viruses like Epstein-Barr virus, EBV.
So HIV was the main focus, but these early gene therapy ideas were being explored for other viruses too.
That's right. The sources mentioned work on similar strategies: antisense, dominant negatives for viruses like human papillomavirus, HPV, herpes simplex virus, HSV, EBV, cytomegalovirus, CMV, trying to disrupt key viral functions by targeting their specific proteins or RNA.
It's actually quite powerful seeing how these really fundamental scientific ideas, messing with RNA, interfering with proteins, engineering immune cells, were being actively pursued decades ago against viruses. It really highlights how current advanced therapies didn't just appear, they built on this long history.
Absolutely. It really underscores that scientific progress is often a long game. Foundational research, even if it doesn't yield immediate cures, paves the way for future breakthroughs, sometimes in ways you couldn't predict back then.
So let's try and bring this all together. Infectious diseases are they're a constant reality, not just some future threat. Climate change, our own activities are significantly dialing up the risk of new ones emerging, mostly through the spillover from animals.
Right.
We've learned some incredibly important, if sometimes painful, lessons about being prepared from recent events. And science is giving us this growing toolkit, a mix of really exciting advanced tech for tracking and diagnosing, built on decades of fundamental research into how these pathogens actually work at the molecular level.
That's a great summary. And it reinforces that preventing the next crisis can't just be about waiting to develop vaccines and tests after an outbreak starts.
It has to be broader.
It has to be. It needs that holistic One Health view. We have to proactively address the underlying drivers, the environmental damage, the biodiversity loss, the social inequalities that basically create the conditions where spillover and rapid spread are more likely to happen in the first place.
It's definitely a complex, interwoven challenge. But understanding these connections, it feels like the essential first step towards actually doing something effective about it.
Indeed. And perhaps it leaves us with this final thought for you, the listener, to consider. We've seen that human activities are really the main thing facilitating spillover. And we've also seen that things beyond traditional healthcare, good governance, social well-being can act as powerful protection against disease. So what if we shifted our focus? What if, instead of primarily reacting to outbreaks, we proactively address these deeper root causes? How might that fundamentally change humanity's relationship with infectious diseases?
That's definitely something worth mulling over long after this deep dive ends. Thank you for joining us today.
Thank you.